Hello, welcome back. This is Professor Young, and this is um, part one of one, looking at exactly how do we propagate a action potential. What is an action potential, um, and how does it relate to what we've been learning in regards to muscle contraction? And really, if you recall, when we started this whole thing on muscles, I said I was going to start the story in the beginning. I'm sorry, I said I was going to start the story in the middle and work our way to the end, and then we would go back to the beginning and begin to fill in the pieces, things that maybe you didn't quite understand. And this is definitely one of those aspects where we're going to go back and look at exactly what is an action potential. And I keep throwing that term around, and I've never really defined it. I've never really gone into any details. And this is where we're going to get to the details. Um, the specific part of the process that we are looking at in regards to muscle contraction is from the time that the acetylcholine opens up the sodium channels to the point in time when this action potential reaches the terminal cistern. And so as it's flowing along the sarcolemma, as that action potential is passing by sodium channel after sodium channel after sodium channel and comes down into the T tubules, that's the part that we're really focusing in on here. How does all of that occur? Some of what we're going to be looking at is going to be new to you. Some of what we're going to be looking at is going to be a review and really just filling in the missing pieces of the conversation and the story. And so to start this off, what I would like to do is revisit this image or concept of a classroom with cats and dogs. And we're going to pretend that cats and dogs are equally positive. Now, we realize that dogs are better than cats, right? But for the argument's sake of our example here, we are going to pretend that dogs and cats are both equal. And in being equal, these are both positive entities. And what we're going to say here is that, what would happen basically if one cat came in and one dog went out, right? Do we really change the overall electrical balance inside of the classroom and outside of the classroom if all we're doing is swapping out one positive for one positive? And the answer is no. A positive cat and a positive dog hold the same merit. They hold the same weight. And so we don't really alter the electrical balance between the hallway and inside of the classroom. Now, what if we, let's see here. What if we bring in one cat and two cat, and we remove one dog, two dog, three dog. So now we've brought in two positive cats, and we've removed three positive dogs into the hallway. Have we altered the balance between the positives between the classroom and the hallway? And the answer is yeah, technically we have. We now have more positives in the hallway than what we do within the classroom. And so in a sense, we can say that that hallway is slightly more positively charged than the classroom. Now, we don't look at this in terms of positives and negatives because we're only focusing on the movement of these positive ions or these positive animals. But in a sense, you could say that the hallway is more positively charged or slightly more positively charged, and the classroom is slightly negatively charged, because it has fewer positive critters in it than the hallway. This little illustration is the basis for our understanding of how an action potential is triggered. It comes down to the movement of ions that sets up one side of the membrane being slightly more positive and the other side of the membrane being slightly more negative. And so let's relate this. In my little example, the dogs equaled sodium ions, the cats equaled potassium ions. The classroom represented the cell. The doorway would have equaled a molecular pump. Specifically, what kind of molecular pump? Well, remember, we got to go back and we got to think about what we're pumping. We are pumping sodium and we are pumping potassium. And so when we talk about what kind of pump are we looking at, we are looking at a sodium potassium pump, which is protein based, and we know that most of our proteins are going to end in ASE. And because this is a pump that is moving ions in a counter current, in other words, it's moving one set of ions in one direction and the other set of the ions in the other direction, therefore this must be requiring energy. And so this is a sodium potassium ATP ACE. That's the specific molecular pump that we are dealing with here. And really what we are, the bottom line is, what we are referring to is an electrical gradient. This is the bottom line. This is what we are creating and generating here. Now, what is an electrical gradient? Well, a gradient is something that changes slowly over time and or distance. Think about the gradient of a road. You go up a very steep hill. That hill can't just move at a 75-degree angle. They've got to keep it at like a 35 or 40, 45-degree angle or less, because otherwise it becomes too steep and some vehicles wouldn't be able to have enough power to get up it. And so that's got to be, that contour has to be a very slow gradient. And so it's a change in something. It's a change over distance or a change over in time that happens very gradually. When we talk about an electrical gradient, we're talking about a change in electrical positioning over a certain distance. In this case, the distance here is inside the membrane and outside of the cell membrane. That's the difference. That's the gradient. All right. It's an electrical gradient because on one side of the membrane we've got a slightly more positive environment, and on the other side of the membrane we have a slightly more negative environment. And so there's a very gradual or slow or minute change in that electrical distribution that happens across the membrane. This concept of having an electrical gradient where one side of the membrane is slightly more positive, in this case, outside of the membrane here is slightly more positive, and the inside is slightly less positive, is known as membrane polarization. We've polarized the sides of the membrane so that one side of the membrane slightly more positive, and the other side of the membrane is slightly more, I'm going to use this term, slightly less positive. That's that's a polarized membrane. Two opposite sides of the spectrum. One slightly more positive, the other side is slightly less positive or slightly more negative. Think about back to the classroom example, moving the cats and the dogs. The hallway had slightly more positive animals in it than what the classroom did. All right? The way that we set up, the way that we create this polarization within the membrane is based on the ability for these molecular pumps to move the ions. And specifically, the ions that they're moving are the potassium and the sodium. And so when we look at what's causing the polarization within the membrane, we need to turn our attention to these molecular pumps. So let's take this to the next step. Let's take this to the next step. Here we have a battery. Right now, we all know that batteries have positive and negative sides to them. That just comes with the territory. That's what they have. Well, we can kind of think of our membrane being polarized as being almost like a battery. Right? What allows a battery to conduct electrical current is the fact that it's polarized. It's got two opposite extremes to it. One extreme is positively charged, and the other extreme is negatively charged. And in fact, if you look in it, and if you take a few seconds, pause the Wimba, count up my sodiums, count up my potassiums, you will see that we have indeed slightly more sodiums here than what we do down here with the potassiums. We have fewer positives down here. We've got more positives up there. It's just what we have. Right? My top side here is positively charged. My negative terminal, technically because it has fewer positives, can be declared negatively, quote unquote, charged, or less positively charged. Another way of looking at membrane polarization is to define it as an unequal distribution, of ions. Isn't that what we have? We have a distribution of positive ions across the membrane, and the positive ions are not distributed equally. One side ends up with more than the other. And that's all we're dealing with. That's all we're talking about when we refer to this thing as this thing known as membrane polarization. When we completely reach and we have true membrane polarization, we have true and complete unequal distribution of these positive charges, known as sodium and potassium. We reach a state known as resting membrane potential. Burn that term into your memory. Resting membrane potential is resting membrane potential is the complete polarization of a cell membrane. All right? Resting membrane potential, RMP, is the complete polarization of a cell membrane. Mathematically, we define that as negative 70 millivolts. Now, this goes back to that exact same thing that we've talked about before. You can't have something be negatively charged. You can't have something that has less than 0 volts. And so that negative that you see plopped in there right there, right, is actually referring to the fact that the inside of the cell is slightly less positively charged than the outside. That's all that negative is referring to. It's referring to the unequal distribution of ions. And it's telling us that the ions are not equally distributed, and in fact, there's more positives outside of the cell than what there is inside of the cell. That's what that negative there means. If it was a positive 70 millivolts, that would tell us that there's more positives inside of the cell membrane than outside of the cell membrane. If there was no denotion there, if there was no positive or negative present, that means it's an equal distribution. And so allow those positive and negatives to tell you where the collection of ions is occurring. That negative tells me that the collection of ions is greater outside than inside the membrane. Make a sense? Right? Hopefully that makes complete sense. Hopefully that makes complete sense to you. Let's take this a step further, if we may. Now, in the PowerPoint, this is an animation. In Wimba, I lose my animation ability. All right? First thing first. A negative 0.07. A negative 0.07 is equal to a negative 70 millivolts. Right? Don't lose sight of that. Please, 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 I beg of you. All right? Don't get thrown off and get all weirded out because it says negative 0.07 volts instead of negative 70 millivolts. It's the same thing. That's number one. I want to throw that out there. Don't freak out on me, please. Number two, what I've done is I've taken the animation that's present within the PowerPoint and I've done screen captures and saved them as JPEGs and inserted them into the presentation here. And what we can do is we can kind of look at what's happening as the ions are moving and how that affects polarization or lack thereof. And so what you're looking at in the first picture, right here, all right, is indeed membrane polarization. Membrane is polarized, okay, which means that then also resting membrane potential is also achieved. And we know that because, check this out, we are at a negative 70 millivolts. We have a greater collection of positive ions outside of the membrane than we do on the inside of the membrane. At some point in time, what we end up having occur is we lose that membrane potential. we lose that membrane potential. In other words, we no longer are polarized. And that's what we see happening in picture 2. All right? In picture 2, what has occurred is all of the sodiums that were at one point in time outside of the membrane have now rushed in to the inside of the membrane. We've now shifted the balance, and we're technically no longer polarized in the sense of we're no longer at resting membrane potential. Now, what we have is we have more positive ions inside than what we do outside. And that is clearly denoted by the change in voltage. We're at positive 30 millivolts. We've gone from a negative 70 millivolts to a positive 30 millivolts. There's now a greater collection of voltage within the cell membrane. There is a lower voltage now outside of the membrane. All right? So that's what's happening here. Now, I will also say this. In picture 2, all right, what we have reached by the opening of the sodium channel, we no longer have polarization. We've surpassed resting membrane potential. And what we do have now is something that is referred to as depolarization. The membrane is depolarized. What causes membrane depolarization? What causes it is very simply acetylcholine. Acetylcholine destabilizes the sodium channels. Remember, that's what opens up the sodium channels to begin with, and it allows the sodium to rush out. And as soon as that sodium rushes out, your voltage changes within the membrane. The membrane's depolarized, and that sends the action potential. And so here's your action potential traveling along the membrane until it reaches the next sodium channel. Now that action potential that reaches the next sodium channel, which by the way is polarized, now depolarizes it. Sodium rushes in, and what does it do? It sends the action potential to the next sodium channel. Yes, action potential sounds like the Jetsons car going through space. And that action potential reaches the next sodium channel, which is polarized. It depolarizes it. Sodium rushes in to the membrane, and the action potential travels on again. And it continues to do that. All right? Now, once that action potential has passed, notice what happens. Notice what happens here. Potassium now rushes out, and we enter into a state of what we call repolarization. Right? Now, repolarization happens in 2 stages. Right? Stage 1 is what you're looking at right here, where we surpass resting membrane potential. And that is a state that is known as hyperpolarization. Hyperpolarization is all about overshooting resting membrane potential. And the reason why the cell membrane would want to overshoot resting membrane potential is for one very simple reason. We need to ensure that we at least reach that negative 70 mil. We need to ensure that we reach it. And by overshooting the mark, we ensure that we reach that negative 70 millivolts. We might not reach resting membrane potential. And the danger of not reaching that resting membrane potential is an accidental firing off of an action potential. And that we don't want to do. Right? That we don't want to do. Because what will happen is, as we will talk about, I think, on the next slide, there is something that is referred to as threshold. Now, this is a concept that we've talked about before. All right, threshold is the minimal amount of energy that is required in order for a reaction to fire off. We have these thresholds within action potential generations as well, and those thresholds are meant to prevent misfirings. Threshold for an action potential is negative 60 millivolts. And so if we didn't overshoot resting membrane potential with hyperpolarization, we might not guarantee that we will reach resting membrane potential. And really, anything under negative 70 millivolts, anything between negative 70 and negative 60 millivolts, is cause for concern for an accidental firing of an action potential, even though we mark that threshold at negative 60. It's kind of a convenience type of thing. So why do we have hyperpolarization? Why do we overshoot resting membrane potential? So we prevent accidentally firing off an action potential by not hitting the mark, by not reaching that negative 70 millivolts. And then what will simply happen is some of that excess potassium diffuses away, and the voltage drops back down to negative 70 millivolts. And so that's what you see happening right there. All right, and at that point in time, in this last picture, we have now reached resting membrane potential once again. All right, and so this here kind of breaks down what I just talked to you about in one nice, neat little slide. We understand that resting membrane potential is defined as being negative 70 millivolts. Once either that acetylcholine or that action potential hits the sodium channel, we hit threshold, sodium rushes in, destabilizes it. In fact, we enter into depolarization, which is also referred to, or which also happens at positive 30 millivolts. We peak out at about positive 50 millivolts, at which point in time the sodium channels close, the potassium channels open, potassium floods out of the cell, all right, and that is where we hit this process of repolarization. And the key here with repolarization, or part of repolarization, is hyperpolarization, where we, again, overshoot the electrical charge. And so we hit, I just called that major typo, negative 80 millivolts. Some of that excess potassium diffuses away, and that drops the electrical charge back down to resting membrane potential. which remember is negative 70 millivolts. And that's where we are. This chart, this table right here, just kind of goes over and looks at exactly what we have already discussed and we've already talked about. Again, just to make sure that we're all on the same page. This location right in here is known as resting membrane potential. Threshold sneaks in right here, and remember that's at negative 60 millivolts. This area right in here is referred to as depolarization. And then we come into this portion of the process right here, which is repolarization. And we also have hyperpolarization, which sneaks in right here. Remember, hyperpolarization kicks in at negative 80 millivolts. And then we return back to, right here, we return back to resting membrane potential, and that is negative 70 millivolts. Right? And so this diagram here kind of graphically explains what it is that we have. And at that point in time, congratulations, woohoo, yay, yippee! You have witnessed an action potential. Congratulations. You are no longer without action potential. You now have action potential. All right, and so that is where we are now. To round out our little discussion here, now that you understand what exactly, why was that acetylcholine critical to the process, and why did I harp so much on the fact that this thing that I abbreviate AP, this thing that we call an action potential, travels along the sarcoplasm down into the T tubules, passing from sodium channel to sodium channel to sodium channel. Now you understand the critical value of that. Now what I want to go back through and do is just kind of fill in some terminology for you, make you sound smart and intelligent when you go through and take AMP2 or run across muscle physiology some other place. And to do that, this here is basically all definition and terminology. Gobbledygook. And that's exactly what we have here. This is pretty much straight terminology. And so as long as you just kind of memorize some terms here, you'll be okay. All right? And the first two terms that we need to consider is a ligand and a receptor. Now, think of this like a lock and key. If you remember back to our discussion on enzymes, we said that the substrate had specificity for an enzyme. In other words, a substrate was specific for a certain enzyme. And that substrates that didn't fit into the enzyme wouldn't get broken down. Well, that same concept is applied here when we talk about neurotransmitters and the channels or the gates that they act upon. When a ligand, which could be a substrate, could be a neurotransmitter, wants to go ahead and act on something, cause a response, it needs to bind into a receptor. If the ligand does not match the receptor the ligand doesn't bind, which means the reaction doesn't happen. So this all needs to be specific. In order to open the sodium channels to start the action potential, a ligand needs to lock into the receptor associated with the sodium channel. That receptor is specific for a certain neurotransmitter. And that neurotransmitter is what we know as acetylcholine. So when we talk about a ligand, an example of a ligand would be acetylcholine. The example of a receptor would be a neurotransmitter receptor that would accept acetylcholine. And so on the sodium channels that produces the action potential, there are receptors that are specific to the neurotransmitter acetylcholine. We see this all the time. You will see this in examples of hormone regulation. Hormones have specific receptors. So hormones can be ligands, and they have specific receptors that they're going to fit into. Substrates are ligands. They have specific receptors that they're going to fit into. Neurotransmitters are ligands. They're going to have specific receptors that they fit into. And as I already mentioned here, acetylcholine is the prime example that we're going to focus on because it directly relates to our discussion on muscle contraction. And again, this here is nothing that we haven't already looked at. This is really the major review, all right, of everything that we've talked about for muscle contraction leading up to the release of calcium from the terminal cisterns. And so we already know acetylcholine is, now we know it as acetylcholine, is going to bind into the receptor that's going to trigger the opening of the sodium channel, which is going to spark, polarize. It's going to depolarize the membrane, which is going to start the action potential and send it down along the sarcolemma. Or, I'm sorry, down along the sarcoplasmic reticulum. Why is it a chemically gated sodium channel? It's chemically gated because there's a chemical reaction that happens. Right? Acetylcholine is going to bind into the receptor and cause a change in the shape. It's chemically induced. Again, this is nothing new. This is nothing that we have not already talked about and looked at in other examples. We're just going to apply it slightly differently. And that is the all or none concept. We're flushing toilets again. That's all we do in this class is flush toilets. All right? We already know that if we expect to leave resting membrane potential and go into depolarization within that cell membrane, that we must, absolute positively, break threshold. Threshold is a guarantee. We need to surpass that. How do we surpass it? By hitting negative 60 millivolts. We don't care if we overshoot over negative 60 millivolts. As long as we reach negative 60, boom, the action potential gets sent. If we don't reach negative 60 millivolts, no action potential. If we hit negative 60 millivolts, action potential goes. That's the basis for sending off that action potential. That's the difference. Negative 60 millivolts is the difference between membrane polarization and membrane depolarization. That's what it comes down to. If you don't reach threshold, nothing else matters. Because you won't have depolarization, you won't have repolarization, you won't have hyperrepolarization, and you won't ever regain resting membrane potential because you would have never left it. let's back up a little bit further. How do we release acetylcholine? Where's acetylcholine even coming from? The answer to that is these gaps that occur between neurons, whether it's a neuron to neuron or whether it's a neuron to a muscle. Now, when we talk about this, the space that is found between a neuron and a muscle cell is what we refer to as a neuroeffector junction. When we talk about the space between a neuron and a neuron, it's a synapse. There is no difference in the structure between a neuron and a neuron and a neuron and a muscle cell. We just change the name. Structure is the same. The interface is the same. All right? It's the same thing. And so very often we just use the term synapse. Neuron or a neuron to a muscle cell. But to be anatomically correct, we use the term neuroeffector junction if we're talking about a neuron to a muscle cell. These last two slides that we are going to consider simply outlines the steps involved in moving the action potential down along the sarcoplasmic reticulum. And so you can see here what happens is the action potential is going to come down the axon of the neuron. Now, the axon is the sending unit of a neuron. We haven't talked about this yet specifically. We will. In fact, our next lecture is going to look at the structure and anatomy of a neuron. And it's going to look at how do we send an action potential down a neuron, because it is different than sending an action potential across the membrane of a muscle cell. It is different. We haven't talked about it yet. We will. So for right now, just know that, yeah, we have action potentials within neurons, and that action potential is going to come down the sending unit known as an axon within the neuron. And it's going to reach the end of the neuron, right? What we call the synaptic cleft or the synaptic end bulb. And within these synaptic end bulbs, we have little tiny vesicles, little tiny chambers that are going to contain a neurotransmitter. Now, in this case, the neurotransmitter of choice is acetylcholine. All right? Now, the challenge here is we've got to open up those vesicles and release the acetylcholine. How do we do that? We do that with our good old friend, calcium. When the action potential reaches the synaptic cleft, the synaptic end bulb, that action potential is an electrical impulse. It is an electrical impulse. And that electrical impulse is going to cause voltage-gated calcium channels to open up and release calcium into the end bulb. The calcium that is now released into the synaptic end bulb is going to bind to the vesicle, and it's going to release the acetylcholine. So make sure you understand this. We're in the neuron. The action potential has traveled down the sending unit of the neuron, known as an axon. And that sparks the opening of a voltage. Action potential is electrical current. it sparks the release or the opening of a voltage-gated calcium channel that releases calcium into that synaptic end bulb. That calcium binds to those vesicles that contain the neurotransmitter. It binds to that vesicle, which forces the vesicle to open, and it releases, in this case, acetylcholine, but it's going to release that neurotransmitter. Once it releases that neurotransmitter diffuses across the cleft, and it's going to bind to a receptor on the other side. Now, in the case of the muscle contraction, acetylcholine is going to be released, it's going to diffuse across that cleft, and it's going to bind to a receptor within the neurolemma of the sarcolemma of the muscle cell. And we know what happens once acetylcholine binds to those receptors within the sarcolemma. We know what happens. Sodium channels are opened. Sodium rushes in. Action potential is sent. And the action potential travels along the sarcoplasmic reticulum and down into the T-tubule, where it reaches the voltage-gated calcium channels. It releases the calcium into the sarcoplasm. From there, the calcium binds to troponin, which is going to spin tropomyosin, which is going to uncover actin. ATP is going to bind to myosin. Myosin binds to actin. ATP releases the phosphate group, creating ATP plus a phosphate, which causes the myosin head to swing, which is going to pull on the actin, which tugs the thin myofilaments, which is going to cause the condensing of the sarcomere, which is going to cause the condensing of the myofibrils, which is going to cause the condensing of the myofiber, leading to a muscle contraction, no more action potentials, so calcium gets reabsorbed back into the terminal cisterns. You know the rest of that story. But in order to get there, the action potential needs to travel down the neuron, reach the synaptic end bulb, cause the release of calcium through the voltage-gated calcium channels. That calcium binds to the vesicles, which then is going to release the neurotransmitter, in this case acetylcholine. Acetylcholine diffuses across the synaptic cleft down into the sarcolemma. Acetylcholine binds to the receptors, and it causes the release of the opening of the sodium channels, which action potential is created. Why is the action potential created? Because you've disrupted polarization, resting membrane potential. And you've depolarized that membrane. And this is just simply a diagram of what all that looks like. All right, and so this here is the synaptic end bulb right there. Right, your little vesicles containing your acetylcholine or your neurotransmitter is right there. Calcium gets released, binds to the synaptic vesicles, and the neurotransmitter gets released and binds to receptors within the tissue. What we refer to as the effector. Right? In this case, it's the muscle tissue. And the rest is history. So I think now you have the complete story from beginning to end. I cannot stress to you enough to review this entire process. You need to be able to explain the entire process from beginning to end, both ends. Right? Both the continuation of contraction and the ending of contraction. There are the two alternative endings that we have to this story. The story starts the same. The middle is the same. It's the ending that can be different. And so with that, I'm going to leave you to studying the complete story of how your muscle contracts. If you have any questions, please, I beg of you, zap me an email, ask me in lecture, ask me in lab, whatever you need to do. Just ask me the questions and get your clarifications. With that happy note, This is Professor Young signing off. Happy studying.